Hello, everyone. Today, thank you for PacBio's invitation to introduce our new technology, HIT-scISoseq, for the single-cell, high-throughput, and high-accuracy single-cell full-length isoform sequencing for epithelial tissues. So my name is Tang, and I'm the Director of Technology, Director of Product, and Director of Research in BGI. And in the same time, I have been the Director of Single-Cell Expression Core in UNR and as Assistant Professor. And I'm very glad to introduce this new technology for you guys today. And so let's start. Actually, we will start from single cell, some basic technology, some basic concept in the single cell part. So single cell, many people think like single cell is like 1 cell, 2 cells, 3 cells, for instance, like that. When I have been the single cell sequencing core director of that core, many of the people think like, okay, the single cell is very hard. The 10 cell is so easy. So, and 100 cell is easier than the 10 cells. Actually, it's not right. Actually, the single cell concept in our field is like it's an increased resolution. It's not like a microscope. So it's not like a microscope can amplify your cells to the level you can see. No, it's just an increased resolution. It's like a camera. It's like a camera from iPhone 1 to iPhone 12. It's like increased pixel. So actually, the resolution is still the same. The resolution get be increased. So it's something like the pixel increase. For instance, like that, you can see in the left side for this Mona Lisa, and you will see it's like a not very clear face, not very clear here, or for instance like that. It's not clear. But for the left side, the Mona Lisa, you will say after the pixel increased, you will say, oh, it's a clear picture. Actually, that's the concept of a single cell. Actually, when you do for the single cell for the whole tissues, you can barely say, okay, for this tissue, you have possibly like here, you have possibly like a nose, eyes, for things like that. It's a whole body, and you can barely say that. But after you make this tissue to single cell levels and you sequence every single cells, and you will say, oh, okay, you can say very clearly, it's nose, it's eyes, it's hairs, for things like that. So actually the single cell technology, actually it's a pixel improvement. It's like a resolution improvement. It's not microscope, okay? So it's not like a microscope can amplify the signal from 1 cell. Yes, it's kind of that, but it's not. It's not that explanation for a single cell. For a single cell, it's pixel improvement. So actually many people think like, okay, the single cell and we have fewer cells, we can do single cell. Actually, it's not right. For single cell experiment, generally, it's like when you have enough amount of tissue and you just want to increase the resolution of these tissues, you do a single cell. It's not like you don't have enough cells and you want to do a single cell. It's not right, okay? So that's the first concept in single cell field. And I want to explain that. And the reason why for that, you will, in this picture, you will see in your left side, here explain method for single cell technology. Actually, the reason why I emphasis on the resolution is because the technology. You will see, actually, it's very easy, and we use oligo-DT with an adapter and do a reverse transcription. And in the other side, it's a technology, and we call template switching, and then we're template switching to the other adapter, and then you will have 2 adapters in the 2 sides, and then you do the PCR. And you do a PCR for single cell, and PCR actually, we do a lot of PCR, and you actually will know in PCR, the bias is very huge, right? So the bias is very huge, and you don't have to mention like you amplify 20,000 different genes. In our library, when you amplify 2 genes, actually the condition is very different. So it's like very different conditions for each gene because the GC is different, the secondary structure is different, the length is different. So actually it's impossible you can get like a linear amplification for every applicant. The standard error for the single cell is very large. So actually that can explain why I say single cell is when you have enough tissues, you just want to increase resolution. Because for 1 cell, this technology is not accurate and the bias is very, very, very huge. So if you want to decrease the bias, the only way to do that is increase the cell number, right? For example, for this tissue in face, you need a lot of cells, like 1,000 cells, to tell you accurately what the expression profile in the face. So you need like over 1,000 cells or over 100 cells. So that's the reason why we say the single cell is increase the resolution. It's not amplify 1 cells. Because when you amplify one cell, you have a great bias. If you want to balance the great bias, you must to do a lot of cells. You must increase the number to balance the single cell bias. So actually, that's the reason why we say single cell is like a big data concept. It's not like a microscope. I repeat this word again and again just to emphasize on the single cell term. It's like high throughput. The reason why it's high throughput, because for each cell it's not accurate. You need a very high number to balance this bias. And so how we do this, how we increase the high throughput, actually all of the single cell, like for example, like 10 times genomics, their single cell technology, and all of the concept is actually based on the droplet-based single-cell-seq. So it's all droplet-based. Why we call it droplet-based? Because we want to do a lot of cells in the single cell, right? So if you want to do one cell, one tube, you possibly need like 10,000 tubes to do that. It's like totally uncontrollable or not doable. So we do this things like we generate a lot of the droplets, and in each droplet we have like one cells in here, and we also have a one microsphere with this cell in one droplet. So this droplet is something, it's kind of a tube. It's kind of a tube in liquid. It's like a droplet tube in the liquid. And when you lysis the cell, and this micro particles will have oligo-DT to capture each RNA on the microparticles. And for the microparticles oligo-DT, oligos, they also have like what we call is here, it's like a cell barcode, where you can see like a cell identification labels, what we call cell barcode. And then we mix these bits together, and then we can do a PCR to amplify that as what we introduced here. But it's worth to mention here is like, as what I say here, you will have a barcode for the oligo-DT, right? You will have a barcode for the oligo-DT. And the barcode is just for the oligo-DT. And in second generation sequencing, and we only can sequence how long? It's like 200 base pair or 400 base pair, right? So you cannot go very long. You just can go 204 and 400. So the only way is we only can see part of the genes close to oligo-DT. So the other term for the single cell sequencing, we call that three-end counting sequencing. So it's a three-end counting sequencing, because for next generation sequencing, we only can see the genes part close to oligo-DT or close to a poly A here, right? Because the barcode is here. So we call that three-end counting sequence. For the single cell sequencing part, actually we only see the gene labels. We cannot see the transcripts labels. We only can see in this gene how much transcript we have for the gene, but we cannot see the transcripts labels. For example, if you have splicing, or you have mutation, or you have say UTR changes, you cannot say that. Okay, it's definitely you cannot say that, and you only can say for the gene labels, not transcript labels. Okay, so actually that's a big basic concept for the single cell sequencing part, and we will quickly go through here, and then we'll go into the new technology we have here. We call full-length single cell sequencing. So for the single cell, where it's useful for three fields. For the first one, you can identify the cell types. And the second one, you can just do the per pseudo analysis. And per pseudo time analysis means you can analyze for the development, for like the cell development, for example, like you can analyze for like the sperm genesis or oocytogenesis, for instance, like that. So, and identify cell types, you can identify different cell types in the tissues, as what I mentioned in the monolysis pictures. And the third one is actually not many people use that. We call reconstruction tissues. And for here, we just will skip that. Okay, let's do a few summary for the single cell technologies. Actually, for the all droplet-based single cell sequencing, include 10 times genomic single cell sequencing. It's all droplet-based. So they have the worth mentioned here is a three-end counting sequencing. It's what very important here. It's three-end counting sequencing, and the throughput is very high, ultra high throughput. And because the throughput is very high and you have very large amount of cells, and in actually the balance we mentioned in PCR will be balanced. So it will be balanced, but it's based on the very high cell numbers. So it's like a big data concept. We are not very accurate for each cells, but when our numbers big enough, and actually we are accurate enough, okay. And you also can put your cells in single tubes, and then you can have many advantages. And the biggest advantage is like you can do single tube, single cells, one tube, one cell, and you can get the full gene coverage because you can label the full genes and to get the full genes coverage, but you have very few cell number, right? You only can do like 100 cells per day. You cannot do 10,000. You cannot do 100,000 cells. So the biggest disadvantage is like it's a very low cell number. The great advantage is like the full gene coverage, because you have only one tube, one cell, and you can label that one cell, right? So you can label that one cell. You don't need to label like the 3 end. You will label the whole transcript in a single cell tube, a single cell, single tube technology. So here is a few summary. But we can think about that. The high throughput sequencing or high throughput single cell sequencing, it's good, but we only can send 3 end. And for single tube, single cell, and actually it's good, but the cell number is too few. Can we balance that? So the best technology for this is if we can use the third generation sequencing, like PacBio Isoseq. If we use the third generation sequencing, we possibly can do that. But in third generation sequencing part, and actually we have many technology here. And why we choose PacBio? The reason why, because nanopore, their accuracy is not enough for us to separate the barcode, because in a single cell, like the 10 times genomics or droplet-based single cell, the barcode is million levels. And actually, if your sequence accuracy is not enough, you cannot separate or you cannot demultiplex the cell barcode, because it's too much and it's too close. Okay, it's too close. But some people use a very long barcode, very, very long barcode to solve this problem. And in a single cell field, and a very long barcode, and you can do nanopore. But the PacBio accuracy is good enough for us to do like the 10 times genomic single cell. They only have 12 to 15 barcode, and but we still can demultiplex that. So we still can separate the barcode to each cells. So it's pretty good. So that's the reason why we choose that. So you will see that actually the whole concept is very similar to what we introduced above. And we just based on the droplets, right? And then we get the full length, and then we just don't do the next generation sequencing. We do the third generation sequencing by the PacBio, and we sequencing barcode, we sequencing UMI, we sequencing oligo-DD, and we sequencing full gene. So after we get the full gene with full transcript, we can actually get the accurate gene structure or accurate transcript structure. So actually, we are stepping the transcript level single cell sequencing. It's not gene level, it's transcript level. So we are stepping the transcript level single cell sequencing. So if we get the transcript level for single cell sequencing, what's the benefit? What can we do for that? So we'll talk about the application first. So what can we do? And then I will talk about some of our technology show our great advance in this new technology. So we will see like in the immunology part, and it has very great advantages and very, very useful. As what we know is like in the immunology part, and we usually talk about TCR and BCR. So you want to know how the antibody get involved and to the antigen, how the antibodies work, how the splicings work. So in actually what we call TCR and BCR sequencing, we want to know that. Actually, now many people publish paper about the TCR and BCR, but can we do that for single cell levels? Because we can match the heavy chain to the light chain, right? If we do the single cell level, we can know more clearly the heavy chain to the light chains pair. Because if you just do the TCR and BCR, you just know the whole population. You cannot not match that. So we want to match that. We want to do a single cell TCR, PCR, but single cell TCR and BCR, as what we know, TCR, PCR is like 1.5 kb around that part, right? And if we just do the next generation sequencing, we do the NGS, and you will say, okay, you only get part of your TCR and BCR, and usually it's 5 part, because people think the high variation area is in the 5 part. But now with the next generation sequencing, we can get the full length. Actually, we have a paper preparing for that. It's like in the next generation, in the PacBio sequencing, we get the full length of the TCR and BCR. And actually, we found that the splicing is a lot in the PCR and BCR, and it's not limited to the 5 end. Actually, it's in the whole body. So we have one paper on the way. And I think that's a very useful part in the single cell full length PacBio sequencing. And the other application is like the mutation and splicing related diseases. Here we give an example like the spinal muscular atrophy, the SM diseases. For this disease, if they have one part like here is a mutation, and the splicing will be changed from here to here. So they will splicing 10, 11, 12 to just 10, 11, okay? So the splicing will be changed based on this mutation. So with this new technology, and you can study some mutation splicing related diseases. That's a second application. For what I am interested in, because I do like developmental biology, and generally I do sperm genesis, and you also can do some oocytogenesis, or for things like that. And for here, the most important things you can facing in a single cell level. What means facing in a single cell level? We know like for each cell, you have two chromosomes, right? Generally, you have two chromosomes. One is from your mother, one is from your father. So we call this maternal transcript and the paternal transcripts. And for the single cell, if you have enough SMP, we call the SMP site to distinguish the maternal transcript to the paternal transcript. And actually, you can know each single cell, how the maternal transcript change, how the paternal transcript change. So in this slide, you will say, okay, so in the very beginning, the maternal transcript will be going to decrease. The paternal transcript may be kept in one level, or they will going to increase, or the zygote micron will going to increase, okay? So actually, that's very useful information when you do developmental biology. So you can distinguish the paternal transcript to the maternal transcript. You can know how these two different transcripts change in the developmental stages. And the other application is like splicing related development. That's what my personal interested in is like sperm genesis. Because in the sperm genesis, they have a lot of the splicing events, like from spermagonia, sperm side to round spermatid and the elongating spermatid. In actually the whole process and the development of the sperm actually usually pair with like splicing. So you have more and more and more splicing, and the transcript will be shortened and shortened and shortened again. That's my personal interest, is like the splicing related development for sperm genesis. So with this new technology, we also can say that how the splicing promote the development. Okay, they're very useful. It's so useful here, but it's a so useful technology. Why we didn't use that? Why until now, and we still not have, we didn't see too many papers around the world. Why? Because they have some difficulties here. The biggest facing difficulty is like our throughput in Sequel, in PacBio platform. Because in the BGI platform, like BGI 2000 or BGI 500 or Illumina, we generally easily to generate like a 400 million reads, right? For easy budget, like for not quite a big money, 400 million reads, it's like a easy budget. But for the Sequel 1 or 2, we only can generate 0.5 to 6 million reads. You know, the 6 million to 400 million, it have 100 difference there, 100-fold difference there, right? So actually it's a big budget. So if you want to do that in Sequel 2, you possibly just spend off like 1 million yuan in the sequencing part for one sample, 1 million yuan, okay? Or it's like $100,000. Okay, it's like a big budget just for one sample sequencing in the Sequel 2. So there's a huge gap between sequencing depths. The price gap is very huge too, as what I mentioned, it's 100-fold, 100-fold difference in the price. And sequence accuracy to debarcoding is also a problem. Even the PacBio platform, it's very accurate. It's so accurate, but still have some distance to the next generation sequencing, right? Because the technology the technology concept is very different. So it still have some way to go. So we also need to solve the problem for debarcoding in the PacBio platform. But it's not that hard because, again, the ISOseq already very accurate. We only just have some technology to help us debarcoding more accurate, but it's already good enough. So how we solve this problem, and we invent a pattern technology. And again, the first several step is the same. It's like generate droplets, and we use 10 times platform or things like that, or other single cell platform like the DropSeq, and then we do the RT reverse transcription, template switching, and get to adapter, and then go to PCR. And the most exciting part, or the most exciting idea here is like how we solve the sequencing depth problem. It's like we link our reads together. we link the reads together because now the IsoSeq already can sequencing the DNA fragment around like 20 KB, 20 kilobase pair. Okay, it's very, very long. But for each transcript in the single cell, it's like around 1.55 kilobase pair. So that means for each spot in the SQL2, we can sequencing, we have enough depth to sequencing 10 different scripts. But in the original designing, we cannot do that. We only can, in each spot, we only can sequencing one transcript, right? So that's original designing. So we change that designing. We link all the reads together, and we can, and then we put the linked reads on the PacBio platform and to sequence that. And we will get the very, very long DNA fragment. And actually, we will get, for each spot, we will get around like 7 transcripts in each spot. That means we can increase the fold, we can increase the sequencing depth to 7-fold. So it's a very great improvement. It's like that can decrease our budget to 10 times. So here's our data. So I will show you some data here, how we get the improvement in the sequencing. You will see for the first, after our pattern technology, we have some pattern here to get some artifacts, because when you link, you have a lot of artifacts here, okay? So you will see the artifacts we can decrease to the very low part. You will see for the normal single cell IsoSeq, for the original designing, we will have 50% of the transcript is what we call artifacts. It's not like a perfect full-length isoform. But after we optimize that, we will get like only around like 10%, close to 10%, possibly 8% artifacts. So for artifacts, we get great improvement. And also for the full length, and you will say we get a very great improvement, like 7-fold, okay? In original IsoSeq, you only can get the mapped full length per minute is like a very few. And after we get the technology improved, we get the fold to 3, and it's like a 6 to 7-fold, the difference. So it's a great improvement in the throughput. So it's great. And then let's take a look at like the correlation. So how about the correlation for our HIT-scISoseq, HIT single cell IsoSeq to the next generation sequencing and per cell barcode? For the cell barcode, did we get any difference? Actually, not too much difference. You will say the linearization is very good. So in actually, our HIT-scISoseq gets a very good linearization for each cell barcode debarcoding. And the debarcoding is very close to the next generation sequencing with our patent technology. We also have a patent in the downstream single cell sequencing debarcoding. We use like the next generation sequencing to help debarcoding in the IsoSeq. So we use some like core debarcoding, patented core debarcoding system to do that. And so you will say, okay, the debarcoding is very close. And then we also say the gene count is different or not. You will say the gene count, majority of the genes have the same have the same expression amount. It's good, but some of the genes not. And actually, that's a difference between next generation sequencing and the third generation sequencing. And actually, the quantification is very, very, very different. As what I said, the next generation sequencing, you quantify the gene levels. But the HIT-scISoseq, we quantify the transcripts levels, and the amplification technology is very different. The NGS, they amplify very short fragment, and the alignment also possibly get some misalignment because we have many very large part is like a not useful part, like oligo-DT or the 3' So they cannot align to the genes. Okay, or they will misalign to genes. But for the full length, you know, the majority part is a useful part, or it's the accurate reading part. You can get the full alignment. So from the experiment PCR part and from the algorithm part, they are all different. So you can see, I think it makes sense. The next generation sequencing and the third generation sequencing, the PacBio, they cannot get a very high correlation. They will get like 0.95. It's already good enough. But for the next generation to the next generation, and you get 0.99, right? But for the next generation sequencing to the IsoSeq, you only can get 0.95. It's already good enough. I think it's pretty good. pretty good figures here. And we also see our correlation between each bench. So we do sample 1 and sample 2. You will see the bench effect is very few. It's good enough, and I think it's a very good technology. And it's very I think it's very stable technology. It's not unstable, because you will see the technology replicate or sample replicate, the correlation is very high, 0.998. Okay. So that's the correlation part. So that makes us believe that it's a very useful technology, or it's a very reliable technology. So for this reliable technology, what can we get from the data? Can we get some beneficials from the data? And can the IsoSeq data totally can replace the NGS or not? If they can replace that, can we get some more extra information for that? So let's see more details about that. So, and after we say, okay, the correlation and linearization is very good, the R square is very good, and so we will see in the application part, how about the correlation between NGS and the IsoSeq. So you will see in the single cell part, what I mentioned, the most useful part is the cell clustering, or which they identify the cell types, right? That's the most useful parts. So you will see in the NGS for this tissue, for this epithelial tissue, it's eye tissue, because I'm not the eye person, but I'm the spermatogenesis person. So for this eye tissue, you will see, okay, we can clustering for 4 different cell types: K15, K4, K12, K14, right? And for the HIT-scISoseq, single cell isoseq, we also can cluster into the 4 different clusters: K15, K12, K40, and K4. So we also can cluster to 4 different clusters. And for each clusters, we also analyze the cluster shared genes, because we want to make sure the cluster we make is the same, right? So we analyze the shared genes in each clusters, and we find, okay, they share 90% of the gene the same. So that means these 2 technologies can cluster in the cell to the same level, to the same format. So it totally can replace the NGS in the single cells. And we also did like a correlation between each cluster, and you will say, okay, for each cluster, there's 2 different technology is very, very close. Okay, it's very, very close. So the correlation is very, 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 very high for each cluster. So for each cell types, for each cell type, it's that matter use NGS and HIT-scISoseq based on the PacBio, and actually the correlation is very high in each cluster. And here's a heat map in each cluster, and you also see this 2 technology. The pattern is very, very similar, right? It's very, very similar. So that means in the cell clustering and the identified cell types level, and this 2 technologies is very close and it's very useful. Okay, so the HIT single cell isoseq can replace the NGS and the single cell field. Okay, but can we get some useful information, or can we get some extra information from the PacBio sequencing, from the isoseq? Because we get the full length, can we get some useful information about that? Okay, we will see here. So actually, that's a very useful information here is like what we find. We find the first thing is like for each cluster, okay, we have some known isoforms. Actually, when you use NGS, you only get here. You only get the white part, okay? You only get the white part, the known isoforms in NGS. So in NGS, you only get the white part, okay? The known isoform, because you only align to a reference in the gene level. You're not aligned to the transcripts level. So you only get the known isoform parts, okay? And we also, by this technology, we can get some known gene novel isoforms. We can get some novel gene isoforms, known, by its known, okay? But the most extent part, we can get some novel gene isoforms. So actually, for each cluster, they have some novel gene isoforms which not classified before. And we also have some gene novel isoforms, known gene isoforms which classified before. So that possibly means for each cell type, they have their unique splicing. Some unique splicing is what we learned before, okay, but it's belong to different cell types. And some unique splicing is what we didn't find before. It's totally novel in each cell type. When you go to the single cell levels, you possibly can say that. When you go to the bulking samples, you possibly cannot say that because you have many abundant transcripts. When you go to single cell levels, you can say that, okay? So we have like a known novel gene isoform and some unknown novel gene isoform here, okay? And we also count the unique isoform in each cell cluster, so in each cell type. And you will say, okay, for each of the cell type, they have like around 2,000 different unique isoforms. That means only this tissue have this isoform. The other tissue don't have this isoform. It's a unique isoform for each cluster. And you will see very clearly, okay? So for each cell type, they will have like around 2,000 different unique isoform, which only belong to this cell type. Okay, okay. For this picture, you will say, okay, here's the unique isoform. Some isoform will be shared between different cells. Some isoform will be unique for each cells, okay? And you will see in this picture too. Here's an example for a very important gene in this tissue is we call CRIT15 genes. And CRIT15, for these genes, you will see they have many different isoform. Documented, you only have two. The documented isoform, you only have two. When we go into a single cell and we find the other 6 different isoforms, and these 6 different isoforms, the abundance is different tissues. You will say it's very different. For this splicing, they're abundant in this tissue. For this splicing, they will abundant for the other tissues. Okay, so for the PacBio, in the PacBio, we can use the isoform to classify the single cell cluster to the other level, what we call transcriptome levels. So today the time is very limited, and you actually have many interesting data didn't show here yet. And for example, we also use the PacBio to cluster the cell types, and we can cluster to more detailed cell types. Usually, you only can use NGS to classify 4 different cell types. But PacBio, you can classify to 8 different cell types. For that data, we didn't demonstrate here, okay? And if you have any question, we will answer your questions in the following Q&A sections. Thank you very much for joining today's webinar.
Hello, good morning, good afternoon, good evening again. It's me. I'm the Director of Marketing for PacBio APAC. And next to me is the presenter who just wrapped up an excellent talk, Dr. Tang Chong, who is the R&D Director at BGI. So Dr. Tong, thanks very much for your excellent talk. And obviously, this is a very hot topic, single cell RNA analysis. So looks like we have attracted a lot of attention. There's many questions came in. So let's address some of those, right? So the first question that came through, what to you is the amount of sequencing depth you want to see for IsoSeq for different applications, single cell bulk transcriptome, for example? Note that the unit of reads for the short reads is very different from long reads, and they're not actually comparable.
Okay, actually, that's a very good question. And for the first question is like the amount of the sequencing depths, and we want to see for IsoSeq for different application. Actually, in my original designing, because like for single cell 10 times cell system, for 10 times single cell system in NGS, and we usually get like 400. We usually get 400 million reads for each run. Okay, so that's normal. And in my original configuration, I think okay, by the I will get like 100 million from the IsoSeq. But in actually by the what we call like the ligation method to increase the throughput, and actually to do 10 IsoSeq. So 10 isoseq 2, your seq2 machine, I need to do 10 of them. So in actually, that's my original planning. But in actually, that is beyond what my expectation is. Like, as what you mentioned, in like the long reads and short reads is not very comparable. So in actually, finally we found we just do one run, we can achieving the same results, because like when you see transcriptomes level, that actually the resolution for each cell is high. So for example, these two cells, their gene expression is similar, but in actually their transcriptome structure is very different. So in actually for the same clustering, like what I show you the picture, and we just do one seq2 isoseq. So we just do one by our ligations method. So it's around like 4 times 7, 42 million reads. So that's enough. And for generally like make this two data comparable, in actually by one nanoseq, the isoseq2 resolution is higher, because actually for data I didn't show you guys here is like we actually by the, if you're sequencing more depths, like do 2 or 3 nan, and you can collect the like 200 million reads by our pattern method, and actually you can get like more clustering than what you can see in NGS single cell. So actually by the same depths, we already can get 20% more clustering than the NGS. For example, like a normal NGS clustering, you can get like a 5 cluster, but by the isoseq, you can get like 7 cluster. So that is what we did in seq1. But now in seq2, it's totally changed the world. And in actually seq2, we just do one seq2, and we can get like by our pattern method, we can get 42 million reads. And actually that is comparable now. Okay, but you want to say what's enough sequencing depths for single cell? There's no enough, because the single cell, even you do the NGS, there's no saturation line. So when you're sequencing more, you will get more. At least like 400 million, 300 million, or 800 million, you can say the saturation. So there is no like the saturation depths. But there's reasonable depths, I will say, by our ligations method, it's one seq2, one isoseq in seq2, and comparable to 400 million reads in NGS. Yeah.
Okay, great. That's very illuminating. The next question came through. It's asking, can we tell apart individual samples from a full-length RNA, like cell hashing that people have done with the 10x technology?
Okay, that's actually a very good question. And actually, we made some problem in this part. And because as what the problem we made is like, actually, we are not very formula, we are not very clear with the 10 times how they tell the cell hashing in the algorithm. So actually, it's very hard for us to just force incorporate the full-length RNA into 10 times that configuration system. So what we did do here is like we're bridging the NGS to we bridge NGS and isoseq together. So we use NGS to configure in 10 times system, and net the 10 times software to configure the cell hashing and the dead cell, the live cell, and for things like that. So the actual 10 times will tell us what the barcode is, like a pure single cell, pure single alive cell. So the 10 times system will tell us, and we use that barcoding, do like a Bayesian configuration. So we do like data Bayesian configuration from the data we got from the NGS, like with the 10x configuration, and we use that as a like a data set, like we did like a Bayesian configuration, and then we can tell apart individual samples from full-length RNA. Yes, we actually here we need to compromise between isoseq and NGS. So that's actually the reason why for that is like the 10 times software is not open source. So actually we don't know what happens there. So for this point, for this time point, we have to bridge them to get like a more comfortable or more reliable results. But in the future, when we